Hello class, and welcome to our next video. In 1.4 we are looking at protein synthesis. So we are looking at both transcription and translation within this, ribosomes, their role, how they are made, both in eukaryotic cells, and we look at a little bit in prokaryotic cells. This video has been broken into two, and there's actually going to be a third one which covers that final point that we've been dying to hear about of our 5' to 3'. Getting straight into it, genes provide instructions for specific proteins. This is not new, and we have learned this previously from our other videos. And so because of that, they do not directly synthesize proteins. Instead, it follows the sequence of DNA, RNA, and then finally our protein synthesis. These sequences of monomers convey information. Depending on the context that we are looking at, we can see that nucleic acids and proteins could be monomers, whereas in DNA and RNA, the monomers are the nitrogenous bases. And then if we're looking at monomers and proteins, we are referring to the amino acids, which can be seen on the table on the right. Forgive me, let's go with my laser pointer. We know how much I like it. The 20 amino acids that are present in all living things. So template encoding strands. This is starting off with transcription. The following video will go into translation. So transcription, the DNA nucleotide sequence of a gene rewritten into RNA nucleotide sequence. So what we can see on the right is our DNA in this bluey green color with yellow, and in the red we see our RNA being transcribed. From our previous video, we know that this RNA that is likely being transcribed would be known as pre-mRNA, and once it has undergone that splicing to extract those exons and remove those introns, we have our mature mRNA form. But for now, we are just focusing on the RNA production from RNA polymerase. So typically mRNA is produced, but there can be other forms as mentioned previously, and these forms are tRNA or rRNA. For each gene, only one of the two strands is transcribed, and then that is why in this image we can see that the RNA appears to be complementary to one side of the DNA strand. This strand that it is complementary to is referred to as the template strand, whereas the other strand will be referred to as the coding strand. And so whilst the coding strand does not directly have anything to do with the RNA, the coding strand, which we can see in this bottom image, does appear to be the exact same as the RNA, except for the replacement of the uracil for thymine, whereas the template strand is the complementary base pairs that would attach to it. So the RNA molecule contains the code for the polypeptide. This polypeptide is what leads us to our proteins. Again, just looking at this image, we can see our DNA strand. We have different names here. We have our non-template strand, or sense, or coding strand, whereas the strand that the mRNA is complementary to is referred to as our template, antisense, or non-coding strand. So this is the transcribed strand. And as we can see, the mRNA or RNA is complementary to the template strand, whereas it is very similar to the coding strand. And so the way I like to think of it is the coding strand mimics the RNA strand, but just with the thymine being replaced with uracil. So the synthesis of this RNA molecule, whether that's mRNA, tRNA, or rRNA, we will focus on tRNA and rRNA later on, but for now the focus is on this mRNA molecule. So this is the carrier of info from template DNA to the protein synthesis machinery. That protein synthesis machinery should be our ribosomes. And what we have first is these recognition sites known as promoters, which come before our transcribed region. This transcribed region is where the DNA polymerase will start to make that mRNA from, but it binds to the promoter site. It binds to this promoter site so it knows where to start making the mRNA. This RNA polymerase unzips the DNA and begins to add the nucleotides. So we say it pries apart DNA and joins together the nucleotides complementary to the template strand. Here we have another image of this, and so we have our RNA polymerase. You do not need to worry about the different numbers of polymerases. Please note that RNA polymerase is different to DNA polymerase because DNA polymerase will only add nucleotides A, T, G, and C, whereas RNA polymerase will add A, U, G, and C. So the RNA polymerase moves along the gene, unwinding the DNA from that promoter section. The newly synthesized RNA molecule is the mRNA. The DNA reforms as the RNA polymerase makes its way along until it reaches a termination site or a stop codon. So we have 3 different stages. There are 40 nucleotides roughly per second in eukaryotes, and a single gene transcribed can be done by multiple RNA polymerase. So the more RNA polymerase we have running along these genes, this leads to more mRNA being produced. More mRNA being produced leads to the increased production of proteins. So here we can see on the top, we have our coding strand. On the top, on the bottom, as the DNA has unwound, we see our template strand. In the orange, we have our RNA being produced, and these are those free nucleotides that get attached by this RNA polymerase. The RNA polymerase will move down the DNA, continuing to make this sequence until it reaches a stop point. This stop point will be a series of genes that terminate the process. Moving on to our genetic code, translation. Whilst this video isn't explicitly on translation, translation is the synthesis of a polypeptide using this genetic code. This genetic info is from the mRNA. So mRNA moved through a ribosome as if a code or a sequence. This code or sequence is translated into amino acids. When we have 3 pairs of nucleotides, this sequence is read as a triplet, and it can be called a codon. And so in this table, this is a codon table. And so you see 3 nucleotides in each box. These 3 nucleotides will make up one of the amino acids. So for our AUG, often our start point, this is methionine, and we have many others that could code for different amino acids. There is only 20 amino acids total, and each of these codons could code for multiple amino acids. So whilst there is, let's have a look at this one in the bottom left corner, we have 4 potential codes that could code for the amino acid VAL. We see AUG in red because this is a start codon, and up here we have 3 in bold that are recognized as our stop. So when the gene is transcribed and we reach this sequence in mRNA, it will stop the process of either making a polypeptide or it will stop the RNA polymerase from running along. The way we read this table is we can start with our first letter. So let's say we have A, our second letter could be C, and finally our third letter, let's say it was U. So we have A, C, U. This is the amino acid that it will code for. So 3 consecutive nucleotides makes an amino acid. There are 64 total codons. So that is every triplet or every 3 pair of nucleotides, and this is our genetic code. It's a very quick video on transcription. Please know that from our last video, it does include those exons, introns, pre-mRNA, and other things like that. Please continue on to the next video to see translation.